In this video, I'm going to introduce fungi and some of the general aspects of the fungal body and life cycle. So fungi are really diverse organisms. Some of them are unicellular, some of them are multicellular. They're widespread throughout the environment, but often invisible to us because, as we'll see, many times they're growing underground where we don't see them. They are absolutely essential for terrestrial ecosystems. One of their most important roles is decomposition. So they are some of the biggest decomposers out there. Oops, there we go. And remember that decomposition is not just aesthetics, getting rid of dead materials or poop and that sort of thing. It's recycling and making nutrients available for new sets of organisms. And so they are quietly and constantly doing this throughout the soil, invisible to us. It's likely that they actually colonized land before plants did. And in fact, they may have been important to establishing a base of soil that plants could survive in, even the earliest plants. And again, that ties back to their major role as decomposers. It's also possible that they played a significant role in making nitrogen available for plants, and nitrogen being an essential compound for plants. Fungi belong to supergroup Opisthoconta, and remember that includes animals and fungi. So within supergroup with Opisthoconta, we have kingdom Animalia and kingdom Fungi. Like plants and animals, they originated in aquatic habitats. And there is a group of protists that are within the supergroup with them, but not part of the kingdom, called Nuclearia, that are their closest relatives. So if I look at supergroup Opisthoconta, I have animals and choanoflagellates. Remember, choanoflagellates are the protists most closely related to animals. And then I have fungi and I have Nuclearia, the protists most closely related to fungi. So because they're in the same supergroup, we know that this suggests that they're closely related to animals. They share a recent common ancestor or a more recent common ancestor than with plants. And this is particularly important. I want to highlight that most people, if you were to ask the general audience, most people assume fungi are closely related to plants. And, I can only speculate on this, but I assume that this ties to movement. People think of fungi as non-moving, growing on, say, the soil of the forest floor. And so they say, oh, fungi must be closely related to plants. But again, remember that is not true. Instead, as humbling as it may be, we are more closely related to a mushroom than to a tree. Some of the ways and evidence that we can see this ties to their nutritional habits. Fungi are also heterotrophic like us. They have to eat. And again, many of them do this by decomposing things that are already dead. Another significant feature is that they store their extra energy as glycogen, whereas plants use starch. All animals also use glycogen. So that's a similarity. So as I was just mentioning, fungi are heterotrophic, and their mode of heterotrophy is more specifically described as absorptive nutrition, which is also what we do. So we make enzymes, and those enzymes take large food, so let me just write this here, large food, and break it down into small pieces, and then we absorb the small pieces. So the enzymes are doing this piece here. And we have a digestive system that's carrying out both of this. So this is all happening inside of our digestive system. And in fact, this part here is happening in the space inside of our intestines. And then from our intestines, they're being absorbed into cells. Fungi have to accomplish this in a different way because they don't have a digestive system. Instead, they secrete exoenzymes. So exo for exit, enzymes that exit the cell, exit the body of the fungi, and go out into the environment. So they actually go out into the environment and digest food outside of the fungi. So it's doing this part here outside of the fungi. And then they absorb those small molecules from the environment. So it's actually digesting the world outside of itself, which is kind of weird. It would be like if we made goo of our food on our plate and then just like laid our cells in the goo and could absorb the goo directly. That's basically what they're doing. And then, as I mentioned before, despite that difference in how we accomplish absorptive nutrition, we all store our energy as glycogen. We also use fat, but our carbohydrate storage is glycogen. And a kind of interesting aside, many very strict vegetarians won't eat mushrooms for this reason, because they bear some similarity to animals. Fungi have cell walls. So again, expanding from 111. In 111, we also often talked about how plants have cell walls and animals don't, but fungi also have cell walls. However, they are made of a material called chitin. If you remember, cell walls of plants are made of cellulose. And so if they're not structurally the same as the cell walls of plants, they're not structurally the same as bacteria. Maybe you remember that bacteria have cell walls made of peptidoglycan. So it's not made of that, it's not made of that, it's made of this material chitin. And if we explore chitin a little bit more, we actually see that chitin becomes a similarity to some animals. Chitin is actually a component of many exoskeletons in insects, for example. I like to say it's the crunch when you step on a bug. So really, even though they have a cell wall and animals don't, the material that their cell wall is made of is a similarity with animals. Because they have cell walls, they cannot engulf food. So they can't go up to like a cell and break it up. So this is why they have to have that absorptive nutrition. So it relates back to their mode of nutrition. It also limits their mobility, right? So in the same way that plants are not flexible and can't move, so too for the fungi. And think about how fragile a mushroom is, right? So if you put your thumbnail against a plant, if you push your thumbnail into a plant or push your thumbnail into a mushroom, you'll damage that tissue. But if you do that to your own skin, while you might leave an imprint, it doesn't actually break the skin. And that's because our tissue has more flexibility. I mentioned earlier that there are many fungi that are unicellular. These are typically called by just a non-technical term, yeast. So these are unicellular. Yeast you may be familiar with is like baker's yeast, but we also use that word for like yeast infections. These are not related to each other in any way. Baker's yeast is Saccharomyces cerevisiae, and yeast infections are an organism called Candida. But what they do have in common is that they're unicellular. Multicellular fungi have a really specific type of body And we'll see a diagram of this in a little bit. But they have their body sort of collectively referred to as a mycelium. So that's like, equal to the whole body. And then they have parts of that mycelium called the hypha. And so to do just like a very simple sketch, this whole thing would be the mycelium. Just one of these parts would be a hypha. Most of the time, the mycelium is invisible to our eyes. So these hypha are tiny, very thin, and microscopic, and they might be running through the soil. So again, this is why they're so invisible to us. The only time we start to see a fungi is when out of that mycelium comes a fruiting body. So this mycelium might be under the ground, so this is the soil, and then at some point it will be triggered to make a fruiting body. And that would be, for example, a mushroom. So a mushroom is one type of fruiting body. So the mushroom only forms part of the year. So you only see mushrooms at a certain time of the year, but underneath that mushroom, so if I were to look under the soil of this mushroom, there's a massive mycelium growing underneath it. The same thing is true with molds. So here on this bread, we see the fruiting body of the mold. That's when you can see it. But before there was ever a fruiting body, the mycelium was growing through the bread. So a lot of times there's already a mycelium in the bread before you ever see any visible mold. And this kind of grosses people out a little bit, right? Because actually, and if you think about it, this is why sometimes bread will start to taste a little bit funny before you actually see any mold. And again, that's because the mycelium is growing through that bread, eating it, right? Breaking it down before you can see it. Then something triggers the fruiting body and you start to see the mold. And the role of the fruiting body is to produce spores. So the fruiting body is involved in reproduction, both asexual and sexual reproduction. So here's a diagram of, from your textbook, of that drawing, so a little nicer than mine. So here is, oops, my pen going here. Here is our mycelium. So this whole thing is the mycelium. These little branches here, they're oftentimes called branches, are the little thin threads running through the soil. This is a second mycelium. And again, there's hypha running through there. And in this case, we're getting a mushroom forming. And so in order for a mushroom to form, you have mushroom sex or fungi sex. And it's not very exciting. The hypha of one, so the hypha of one type of mycelium and the other, they fuse with each other somewhere where they touch. And out of that fusion will grow the fruiting body. And so again, most of the time, this is kind of the invisible life of the fungus. And then just every now and then, it makes this fruiting body. So when mycelia are growing, whether it's in the soil or it's the fruiting body, they grow quickly when food is plentiful. So like that piece of bread, yum, yum, they're eating that bread as they move through it, and they can move through it very quickly. And as you may be familiar with at home, the warmer it is, the faster that will happen. They grow at the edges, so if you think about the mycelium, they're growing here at these tips. And as they grow like this, they make lots of branch points. And so even though their bodies are really fragile and thin, by having all of these branch points, they actually have a very high surface area for absorption. For those of you who've taken a little bit of physiology, you may know that we have something similar going on in our intestines, where we have structures kind of shaped like this that optimize surface area for absorption from our intestines too. Same basic idea. That high surface area lets lots of nutrients pass through. It also allows water movement. And this is how fungi grow, which is why fungi are also limited by water. So you will see abundant fungal growth with moisture, but dry soil has a lot less fungal growth. And again, if you think about that bread, right, same thing. If you have dry bread, you get no growth, right? So if your bread has desiccated, which, happens a lot in Colorado, like your French bread, it's not going to grow mold on it. It's just going to be all desiccated, because even if there was mold spores there, they're not able to expand. So this is taking a closer look at that process. So this is the tip of a hypha. So again, this is where the growth is occurring. And so water flows in, and as it flows in, it swells the cell, and that allows it to move this direction. So to match that swelling, it's forming new cell wall, right? So that's why it doesn't just burst, for example. So to match that water swelling, it makes some cell wall. And then the other things we see happening here is a feeding process. So here's digestive enzymes, those exoenzymes going out into the environment. And as they break down the food into small pieces, the food pieces enter into that cell. And again, they're expanding rapidly when there's a lot of food available and moisture available. So if you're not familiar, right, with molds, which I doubt anyone isn't, right, this is a picture of some mycelia growing, in this case in culture. So this is in a liquid culture. This is on a agar plate, kind of like how we grew bacteria. And remember how some of our plates actually got mold contamination on them. In nature, mycelia are usually pretty irregular. So again, just kind of these random branching. In liquid culture, they oftentimes take on a spherical shape. So this is like a ball of mold. I don't know if this ever happens at your house, but I accidentally make these in juice sometimes because there's an abundance of stuff in there, and if the mold happens to get in there, they can grow this way. We're getting a circular shape of the mold here as well, but it's flat because it's on that agar plate. But again, in nature, it's oftentimes more of a asymmetrical, more natural process. Some of the fungi differ in how their cells are divided. Across the mycelium. It's not significant for phylogeny, as it turns out. They used to think it was, and so fungi are often described based on these terms we're going to see here. And remember, that means it doesn't tell us about evolutionary relationships. But again, like we've said many times, even though it turns out that it's not important for evolutionary relationships, it's a structural feature of the organisms that is we're able to determine and see. And so people still like to talk about this trait. So the first one here is septate fungi. And so what we see here is cross walls. So this is a hypha. This whole thing is a hyphal branch. And each one of these dots is a nucleus. And you see these cross walls between the nuclei. So the whole thing is surrounded by cell wall. That's that kind of little purplish color. And then you have this cross wall in between the nuclei. Notice there's a little bit of a gap in the wall that allows the movement of materials. And so septate means separated. They're separated by that cross wall. Some fungi are aseptate. That means unseparated. So aseptate means without a separation. And so again, here's our nuclei, but notice there's no cross wall. This is also sometimes described as being multinucleate. Again, for those of you who maybe started to take physiology, we have cells that are like this, like our muscle cells are multinucleate without division. So again, there's just that cross wall would look like this, and we don't have that. So they're not partitioned into smaller cell walls. And so the significance of this, again, if you remember back to 111, is that you don't have to do cytokinesis after mitosis. Okay. So they still do mitosis to make more nuclei and grow, but they don't have to put in a new wall or new membrane. And again, this is a trait that we can describe about fungi, even though it's not important for phylogeny. So we'll introduce reproduction here in this section as well, but we will also talk about reproduction at greater length when we get into talking about fungal groups, because reproduction varies across the fungal groups. So we'll make some generalizations in this part, and then when we talk about fungal phylums, we'll talk about how they differ from each other. So many fungi use both asexual and sexual reproduction. Some only reproduce asexually, okay? So there's no fungus that only reproduces sexually, though. So some are only asexual, some are both, okay? And this is an example of an asexually reproducing fungus here, and it's still producing spores. So regardless of what is happening, asexual or sexual, it will always produce spores. So we always call the reproductive cells spores. They'll have different names. So conidia is one type of asexual spore. And what basically happens is the mycelia just produce them in a special area. And it's through mitosis, because the mycelia is haploid, and it will just produce haploid spores by mitosis at the tips of branches. And so what you have here is a hyphal tip, and you have mitosis producing asexual spores without any contribution from two parents. In sexual reproduction, it's going to involve processes that are familiar to us. So we have gametes, a zygote, and meiosis that yield sexual spores. So as I mentioned a moment ago, the mycelium is always haploid. So when you have hypha here, so this is one mycelium, this is another mycelium. We oftentimes call them positive and negative mating types. They differ biochemically, but not structurally. This is one N, this is one N. So when they fuse, where they meet, that produces a diploid cell. And then what actually a lot of times happens in fungi is that they're more specifically referred to as heterokaryotic. So I'm going to write that as two nuclei that remain separate instead of fusing. So come over here, it would be a cell that has one nuclei and another nuclei. This came from the positive mating type, this came from the negative type. So there's two nuclei there, right? So that's why we can also sort of think of them as 2N, but they remain separate. And then as they divide, so I'll put two fungal cells there, they keep those separate nuclei. So positive, negative, positive, negative. So as the fruiting body forms, it's making this heterokaryotic structure. So every single cell inside of this fruiting body has a positive and a negative nuclei. And then somewhere up in the structure where the spores are going to be produced, then you get fusion of the nuclei. So this is fusion of the branches. That's what leads to this structure. But then somewhere within this structure, you get fruiting of the actual nuclei. Once you get fusion of the nuclei, so this is where I'm at here, you go through meiosis. So now you have 2N. You go through meiosis and go back to making haploid spores. But because you had the positive and negative mating types, these spores are now unique compared to asexual spores. The fruiting bodies are going to differ in their structures, and again, this is going to have to do with which group they belong to, but it's also going to have to do with who helps disperse the spores. So some spores will be wind dispersed, some will need rain to be dispersed, some will use animals to be dispersed. In this case, this is called a stinkhorn, this one over here. And the name kind of gives away that it's a little bit smelly, and insects are attracted to it. And so when they crawl over the stinkhorn, they move the spores around. So this is the general sexual life cycle of fungi. And again, we'll look at more specific ones when we talk about the phylum or the fungal phyla. So they can asexually reproduce too. So here's the mycelium. And so at the tips of their mycelial branches, or the hyphal branches, they can just make structures that will produce spores through mitosis. So they don't have to do anything special, just through mitosis, because this is already haploid, going to make haploid spores. Those spores can germinate and make new mycelia. And this is what a lot of fungi do a lot of the time, especially molds. However, sometimes they can get hyphal branches. So there's one branch. There's another branch. We have our positive and negative type. These guys can grow together and get fusion of the cytoplasm. So where they fuse, so after fusion, they now have the positive and negative nuclei. That's what gives this heterokaryotic stage. And then that will start growing into heterokaryotic mycelia. And this is what produces the fruiting body. So the whole fruiting body is heterokaryotic as it grows through mitosis. So it's maintaining those multiple nuclei. Within the fruiting body, so here's my mushroom. So here in a specific part of the mushroom, like for example, the gills, I will get fusion of the nuclei. So those two different nuclei come together to make, instead of having two different ones, to make one that's truly 2N. And then once that happens, it will go through meiosis. So that diploid cell will go through meiosis and produce the haploid spores, but these are now unique, whereas our asexual spores were identical. So there's my haploid unique spores that can also germinate and produce a new mycelium. And that's the general life cycle. And again, we'll see variation in it based on the type of fungi, but that's the basic idea. And so here's a picture of that from your textbook. So here's the fusion. So positive and negative mating types, they fuse and form heterokaryotic, and we can say dikaryotic too, that's just for two. So it's about equivalent to heterokaryotic. There's a subtle difference that we don't need to talk about. So there they are, and you can see that this is going through mitosis and making more cells that are heterokaryotic. So red and blue, red and blue, red and blue in each one of those cells. And that's what's happening inside of this fruiting body. This part is still the regular haploid tissue, but this whole thing is heterokaryotic. And then somewhere inside of here, that's where we're going to get meiosis and production of spores. And then each of those spores can go and germinate a new offspring. So I want to wrap up this section by making a chart that summarizes some of the traits of plants, fungi, and animals. So I'm going to make a big table here. And doing my best to be tidy. It's a little hard with this pen. Oops. All right, so I'm just going to put in some traits here. So we've got plants, fungi, animals, and just to remind you, these are both in supergroup Opisthoconta. This is in supergroup land plants and algal relatives. So motility in plants, no, they're non-modal. Fungi are non-modal. Animals are generally modal. We'll talk about exceptions to this, but we'll go ahead and say generally yes. Nutrition. Plants are autotrophs. Fungi have to eat, and so do animals. And in fact, we can also say more specifically that they use that absorptive nutrition. Cell wall. So again, yes, but it's made of cellulose. Fungi have a yes, but it's made of chitin. And animals are a big no. Okay. So for carb energy storage, plants rely on the molecule starch as their energy storage carbohydrate, whereas both fungi and animals use glycogen. If we look at the bodies of these organisms, we see some differences. So plants are multicellular. Fungi can be unicellular or multicellular, and animals are multicellular. So here's a place where we see more similarity between plants and animals. If we look at that multicellular tissue, so get a little more fine-grained into what that tissue is like. Remember, plants have thick three-dimensional bodies in that multicellular tissue. So do animals. We have thick three-dimensional bodies as well. If you think about fungi, though, they have that simple filamentous structure. And that means one layer. So let me just come over here for a second. So their branches are like just, so we saw their mycelium, right? They have these branches. And again, these are super thin. You can't even see them with the naked eye. But even the fruiting body, so if I come over to this fruiting body here, if I look at that fruiting body, it's not like our tissues where it's complexly layered with multiple different types of cells. It's really just taken hyphal branches and stacked them. So rather than being truly three-dimensional complex tissues, it's just those single layers stacked. So it's like pieces of paper stacked on top of each other, not a, not a book with like a cover and a thicker front page and a thinner front page, right? It's just exactly the same pieces of paper stacked on top of each other. So it's much simpler tissue type than what we see in plants and animals.